part 2 of the immunotherapy in bioregenerative medicine, we talk about existing modalities targeting immunosenescence, starting with nutrition, macro and micronutrients. Authors of this book present data regarding influence of various factors on the occurrence and progression of the immunosenescence. For instance, such as correlation between metabolic syndrome, obesity and diabetes and immunosenescence, neurodegeneration, atherosclerosis, response to vaccinations, and so on and so forth. In the part 2 of the immunotherapy in bioregenerative medicine, we talk about existing modalities targeting immunosenescence, starting with nutrition, macro and micronutrients. The main goal of nutritional immunology is to study the effects of nutrients on the immune system in details. Suboptimal nutrition, characterized by micronutrient and macronutrient deficiencies, is typical in elderly individuals and can lead to ineffective immune responses and cause vulnerability to pathogens. Nutrients themselves can act on immunosenescence process and mediate pro- or anti-inflammatory responses, influence inflammation mechanisms. Apart from essential nutritional elements, a healthy diet includes 3 important sources of bioactive ingredients: phytomolecules, probiotic bacteria, and omega-3 fatty acids. Approximately 40 micronutrients, such as vitamins, essential minerals, and other compounds required in small amount for proper metabolism, have been reported as essential dietary components. In humans, fasting or caloric restriction modulates several molecular signaling pathways that maintain energy activity in aging T lymphocytes. This is primarily because nutrient scarcity and energy deficiency activate AMPK-activated protein kinase, AMPK, which promotes mitochondrial regeneration through the expression of the nuclear respiratory factor NRF1 and NRF2. In turn, this stimulates antioxidant response elements, which inhibit deleterious effects of free radicals on the immune system. However, chronic caloric restriction may also impair some immune functions, possibly through inhibition of the mammalian target of rapamycin, the mTOR, although this side effect of caloric restriction may be limited by alternating cycles of caloric restriction with a standard diet. In fact, either fasting, only water for 48 hours, or the fast metabolism diet, a diet low in protein and sugar and high in fat but low in calories, lasting for 2 to 5 days and alternating with longer periods on a standard diet, can be effective and in reversing markers of immunodeficiency. Several studies have reported the immunomodulatory effects of nutraceuticals in aging. These studies have shown that secondary bioactive metabolites confer pharmacological actions against immunosenescence. Polyphenols such as EGCG, quercetin, resveratrol, cysteine, and myricetin have shown potential as a reliever of stress-induced cellular senescence development, replicative senescent-associated secretory phenotype, as well as senolytic attributes in various experimental settings. The polyphenol-rich fraction of Azarella compacta, activated human NK cells and T cells, has been shown to act as a non-specific immune modulator during human immunostimulation in vitro. Another study has shown that grape polyphenols enhance the chemokine functional response of neutrophils isolated from young and old human subjects by altering CD16 and CD66B expression. The profound research done by the scientific research team of European Wellness Group and MF3, MF+, translates into the successful formulation of the highly effective products, health supplements, soft gels with nutrients of placenta, sublingual preparations such as the liposomal Swiss Natural range, mitochondrial peptides, micro-organelles, nanopeptides, non-organopeptides, and many others. In addition to photochemical metabolites, there is increasing evidence of the positive effects of probiotics or symbiotics on the composition of the gut microbiome and protection against immunosenescence. For that, MF3 also has created a therapeutic solution, the symbiotic probiotic. The colon is populated with more than 1 × 10^14 bacterial cells from several lineages, in particular firmicutes, the bacteroids, actinobacteria, proteobacteria, of which firmicutes and bacteroides predominate. The microbiota is highly variable between individuals and comprises of approximately 55 species. Disruptions in this balance and the resulting dysbiosis are often associated with inflammation and poor barrier function, allowing constituents of the microbiota to potentially cause significant disease to translocate the epithelium and spread and infect other distant sites of the bodies, practically metastasize, the bacterial metastasize, and with potentially catastrophic and fatal consequences. The gut microbiota is established in a parallel with the immune system. The immune system is functionally immature in newborns who become immunocompetent and able to mount effective T and B cell responses as the gut microbiota become established. An important factor in the efficient development of both the gut microbiota and the immune system in early nutrition, including the positive effect that breastfeeding and breast milk have in helping to establish beneficial bifidobacteria, which can modulate the immune system and its responses. Aging affects the resident gut microbes with a reduction in the proportion of Bacteroides, Clostridia, and total lactobacilli population, in parallel with increased in Fusobacteria, Streptococci, and Staphylococci. Interestingly, studies have shown that a lower species diversity and a reduction in bifidobacteria in fecal samples from elderly individuals who are aged between 67 to 88 years of age correspond to altered functionality and reduced ability of bifidobacterial strains from elderly individuals to adhere to intestinal mucosa. This chapter also highlights all essential micronutrients and vitamins in regards to their association to immunosenescence.